Morning, Miss George.
Good morning, Mr. Metzger. I heard you missed my class because you overslept.
Yeah, I was doing the social studies project all night.
Well, thank God for study you all. Your assignment's next to the microscope.
Alright, I see it. Alright, I got this. Oh, yeah, what's up, by the way? Alright.
Hey.
What? I love science. This can't be happening. Come on.
I'm sorry, I didn't mean to scare you. I should formally introduce myself and my fellow organelles. I'm Melanie the mitochondria, and I break down energy and store it for later. Over there is my friend Ronnie the ribose, and he creates protein. This is Corey the chloroplast, and he uses sun energy to create food. This is Gloria the googly body, and she packages and delivers material. This is Edward the ER, and he moves material. This is Ned the nucleus, and he contains genetic information, is the site of protein synthesis, and assembles ribosomes. You know, introducing you to all my friends just isn't enough. I need you to come in the cell.
Where am I?
You're in a plant cell.
Plant cell. Looks like a house.
Well, where else do you think us organelles live?
Fair point. But, why is it decorated like Christmas in here?
'Tis the season, but that's besides the point. I want to give you a tour.
Oh, tour. Sounds nice.
Yeah, I'm going to teach you everything there is to know about a cell.
Everything?
Everything.
Good, because I'm doing rough and by right now, and I'm gonna get kicked off the basketball team if I don't get that grade up.
Well, so to start the tour, we're going to look at the 7 scientists that helped develop the modern cell theory. This is Robert Hooke, and he used a compound light microscope to observe a piece of cork, and he found geometric shapes, which he called cells. This is Van Leeuwenhoek, and he became the first man to observe a single-celled organism.
Oh, nice haircut.
And both of these two men together, created the first part of the cell theory, which is all living things are made up of cells. And then these 2 scientists helped discover the second part of cell theory, being that cells are the building blocks of all living things. This is Matthias Sheldon, and he observed plant tissue and saw that it was made up of cells. This is Schwann, and he observed animal tissue and discovered that it was made up out of cells.
What did you say this guy?
These last 3 men came up with the third and final part of cell theory, being that cells can't spontaneously generate. They have to be made from pre-existing cells. This is Francisco Redi, and he conducted an experiment that disproved the myth that maggots form on meat.
I like his haircut even more.
This is Pasteur, and he conducted an experiment that shows sitting broth can't have microbiological life within it.
He looks like my uncle.
And this is Virchow, and he made the discovery that cells come from pre-existing cells.
Looks like.
This is our arch nemesis, the prokaryotic cell. They're very envious of us eukaryotic cells because they don't have membrane-bound structures.
How am I supposed to remember all this?
You just have to try. You might have noticed all these red spheres hanging around. They're the hydrophilic heads of a phospholipid bilayer.
Yeah, they're fun to play with.
Well, the hydrophilic heads love water, unlike the hydrophobic heads, which despise water. This is the energiometer, and it measures the amount of energy in the cell.
Where does the energy come from, though?
ATP.
ATP.
ATP is adenosine triphosphate. It's made up of an adenosine, a ribose sugar, and 3 phosphate groups. And when the bond between the last 2 phosphate groups are broken, energy is released, and a machine in the cell captures it and puts it in energy storage, and it's trapped here.
Kind of sort of makes sense.
We wouldn't want it to run too low, or else we wouldn't be able to do everyday activities. Hey look, it's Corey.
Howdy, y'all. How do you do, man?
Hi, how are you? Corey is a chloroplast, and he's kind of special. See, photosynthesis is constantly taking place in Corey, and photosynthesis is split up into 2 main parts, the light-dependent reactions and the Calvin cycle. The light-dependent reaction occurs in the thylakoid membrane, which is Corey's right arm. The light-dependent reaction uses water and sunlight to create oxygen, which is released in the atmosphere. It also creates NADPH and ATP, which is then used in the Calvin cycle. The Calvin cycle is on his left leg, and it occurs in the stroma. The Calvin cycle uses carbon dioxide to create glucose. It also creates NADP plus and ADP, which is then used in the light-dependent cycle. One of the stages of cellular respiration is the Krebs cycle. The Krebs cycle occurs in the matrix of the mitochondria. The Krebs cycle needs oxygen in order to create 4 carbon dioxides and 2 ATPs. The Krebs cycle turns twice per glucose molecule.
Twice, you say?
Twice.
Alright.
Another stage of cellular respiration is the electron transport chain. The electron transport chain occurs in the inner membrane of the mitochondria, which is about here. The electron transport chain requires FADH, NADH, a hydrogen ion, and oxygen in order to create 34 ATP. There's one other stage of cellular respiration, but it doesn't occur in the mitochondria. It occurs in the cytoplasm, which is everywhere around us. This phase is glycolysis. Glycolysis uses glucose and ATP to produce 2 ATPs, proteases, hydrogen ions, and electrons.
Oh, so there's 3.
Yes. So this is our cellular transport room, and it mimics what cellular transpiration is. What cellular transpiration is the movement of molecules across the membrane. Before we enter, we have to pick what type of transport we want. Osmosis, active transport, facilitated diffusion, or passive transport.
Well, what do those mean?
Well, osmosis is the diffusion of water. Passive transport is the diffusion of molecules from a concentration of high to a concentration of low, and it does not use energy.
So they're kind of the same?
Kind of, but osmosis is water. Active transport goes from a low concentration to a high concentration, and it does not use energy. And facilitated diffusion is very similar to passive transport because it does not use energy, but it requires a transport protein to get a molecule across a membrane. This is a hypotonic solution. Notice how there are more water molecules outside of the cell than inside the cell. This causes the water to diffuse through the membrane into the cell, which causes the death of the cell from bursting, which is called cytolysis. This is a hypertonic solution. Notice how there is more water inside the cell than outside of the cell. This causes the water to rush out, causing the cell to die through the process of plasmolysis. This is an isotonic solution. There's equal amount of water inside and outside. The water will move back and forth through the cell membrane, but will have no result. What else is there to tell you about a cell? I got it.
Well, what is it?
Fermentation.
Oh, what is fermentation?
There's 2 types of fermentation, alcoholic fermentation and lactic acid fermentation. Lactic acid fermentation occurs in the cytoplasm. It requires glucose in order to form citric acid, ATP, and heat. The second type of fermentation is alcoholic fermentation.
Oh.
Do you have a question?
Yeah, what's fermentation?
That's what I'm explaining.
Okay, continue then.
Alcoholic fermentation occurs in the walls, the cell walls, and it requires glucose and ATP in order to create ethanol, ATP, carbon dioxide, and heat. I think this ends our tour.
Wow, alright. I've never had so much fun learning before, like, honestly. This was fun. What's that?
It was mitosis. It's about to occur.
What's mitosis?
It's the phase after interphase.
What is interphase?
You know, Trey, cells can't live forever, so they have to go through a process of mitosis, which is the splitting of a cell.
Oh.
But before the actual splitting of a cell, the cell must go through interphase. Interphase consists of 3 stages, G1, S, and G2. G1 and G2 are the growth and development phases, and S is when chromosomes replicate. After interphase comes mitosis, which is broken up into 4 parts: prophase, metaphase, anaphase, and telophase. Prophase is the first part, where doubled chromosomes form, the nuclear envelope disappears, and a spindle forms. So then comes metaphase, where the chromosomes align in the middle of the spindle.
Yep.
Then it's anaphase, where the chromosomes split in half and the single chromosomes move to opposite sides of the spindles. And then lastly is telophase,
Yep.
where the spindles disappear and nuclear envelopes form over the different sets of chromosomes.
That's a lot.
Very much a lot.
After mitosis?
Yeah, the actual splitting of the cell is called cytokinesis. It's when a plate forms in the middle of the cell, and it's the splitting of the cytoplasm. And we have about 5 minutes until this cell splits, and if you don't leave now, you're going to be stuck in here forever.
Well, how do I go home?
You just have to wish.
Alright, well, before I do, I just want to say thank you for all your help.
Oh, thank you. You're going to do so good on that bio lab.
I hope so.
Digga 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 digga. Oh.
You have 5 minutes, okay, hon?
I got you. I know all of these. Oh, sweet, my bio grade's in. Thank you, Melina. I got a 100.